Yeah, next we have our local hero, Lars Velten, and he'll be telling us about cell-state-specific enhancers in hematopoiesis.
Yeah, thanks, Johnny, for the introduction and for this nice invitation to speak here. We will switch gears a little bit now, because now we're getting really deep into non-regulatory biology. And we're doing this in a specific system, but the system, I think, for you guys is less relevant. I still want to introduce it. My group is exclusively working on platformation, which is a system where every day 1 trillion new white and red blood cells need to be generated in each of your bodies. And ultimately, all of these cells derive from stem cells. Of stem cells, we have very few. We have 100,000 of them. They have to serve us all our lives, and they are usually not doing anything. Question we face in the community working on this biological system is that during the differentiation of these stem cells, transcription factors, master regulatory transcription factors, are expressed in very overlapping gradients. And I think the same is true for many developmental systems. However, the downstream target genes of these transcription factors are expressed much more stage specifically. So basically the question we're after is how can regulatory DNA encode specificity given that the input that the regulatory DNA receives doesn't seem to be very specific, right? So there need to be some like nonlinear interactions here. And the big challenge in understanding regulatory DNA is this issue of combinatorial complexity, which I think was beautifully described in this opinion piece by Karl de Boer and Jussi Taipale, who basically say transcription factors can interact in really many ways. And if you do the maths, the human genome has over 1,600 transcription factors. Their interaction is known to depend on the spacing between them and the orientation between them. So basically, for even just pairwise transcription factor interactions, you have like on the order of 220 million transcription factor cooperativity unknowns. And basically to cover each of these possibilities once in a perfect training data set, you would need 6 gigabases of DNA. But the human genome has only 3 gigabases, and it's really not designed for learning the code of gene regulation. So basically, we're facing a shortage of training data. And the idea that Karl and Jussi are proposing is to work with random sequences and hold out the genome as a test set, which I think from a machine learning perspective is quite cool. So they did this in yeast. This is really not my work. I'm just getting started. But they did this in yeast. They trained, I think their earlier paper was with 10 to the power of 8, 100 million random sequences. And here's the test set, the performance of the model they trained on the test set being the natural genome. And performance is pretty impressive. However, this is the mean activity level in the yeast natural genome. And I think in regulatory DNA, what we're actually interested in is specificity. So they did apply this to switch of carbon source, which I think is the closest that yeast has to different tissues, right? But what we really want to do is like understand cell state specificity in mammalian genomes. And I would argue that it's like much more exciting trying to understand the difference between, let's say, a white blood cell precursor and a different type of white blood cell precursor as compared to a kidney cell and a neuron. Because obviously, we want to achieve specificity during developmental transitions. So to tackle this question, this was basically the big project in my lab for the past 5 years. We put together what I now think was quite a dream team. My first PhD student, Robert, who has already left, supported by our 2 technicians, Chelsea and Aina. The second PhD student, Julia, who's been specifically focusing on technology development. She's actually here, and I heard soon looking for postdocs. And then on the TriLab side, Felix, PhD student, and a collaborator from the collaboratorium Bosa. So what have we done? We said, okay, just working with random DNA in the context of a mammalian differentiation system is not going to be feasible for experimental reasons. And so we decided to focus on a small fraction of this huge transcription factor universe. And we picked our 38 favorite transcription factors. And then we made random DNA. So each of our construct has a different random DNA background, and I think this is essential because otherwise you're learning like in one constant background, and you don't want to do that. So we took random DNA, many different pieces of random DNA, and placed binding sites for these transcription factors in the random DNA in different repeats, different affinities, spacings between them, orientations. We did this for one factor at a time. Then we chose 10 transcription factors and extensively explored all pairwise interactions between these factors. And finally, we did a less finely sampled survey of all pairwise interactions between 42 pair factors, with then only 27 sequences per factor combination, so exploring less options on how they can be spaced and so on. So this is basically our training data. And, well, I already stressed that we have different random DNA backgrounds. So what is our assay? Our assay is so-called massively parallel reporter assay. I know many of you are working on coding sequences, so I should briefly explain this. An MPRA is an assay where you clone in all these synthetic DNA sequences, and this drives expression of a barcoded GFP from a minimal promoter. What is important is that, of course, we did this in the cell state specific, in a cell differentiation system doing cell state specific readouts. So we have this culture system for hematopoietic stem cells, and we then take out 7 different cell states and quantify the activity of these constructs in each of these cell states. I'll structure my talk into 3 bits. The first one is very biological and qualitative. It mostly serves to show you how complex transcriptional regulation really is. A bit of a technical thing, we have lots of random DNA controls in our library, so we quantify for each transcription factor to what extent it creates, the presence of its binding sites creates sequences that have activity that is higher than that of random DNA, or in some cases also to what extent it represses transcription below the baseline level of random DNA. And we do this for each group of sequences defined by presence of a transcription factor, and we then get these maps where we see so here are the single factors, here are more single factors, these are the cell states, and we basically see whether these transcription factors are activators, such as here, activators in all the cell states, repressors here specific to some cell states. And what you can see just by coarsely looking at this map is that the single factor enhancers, so enhancers that contain really binding sites for a single factor, they are not very specific. You mostly get activity like all over the place. However, if we take pairs of transcription factors, you get more specificity in terms of activity, and you get certain combinations that in some cell states are activators and in others are repressors. This is quite interesting, and I will come back to this, of course. But just glancing at the data, the message here is that with enhancers that only contain binding sites for single transcription factors, of course, do not achieve specificity. How could they if the factors themselves are expressed in a non-specific manner? However, as soon as we go to pairs, we achieve specificity, and these cell states then cluster to the known transcriptional and developmental relationships between them, so stem cells sort of as an outgroup, and then the 2 major branches of the hematopoietic system. Okay, so that's like a very coarse look at the data. Now diving in, something very simple we can do is we can correlate transcription factor expression to the activity of enhancers that contain, well, just that binding site, and this basically serves us to, is useful to confirm that, yes, some of these factors are activators, some of these factors are repressors. We can also check how many motifs we've now placed and how good those motifs are. So we agglomerate these 2 quantities into something we call a sum of motive scores, which basically expresses whether I have here like 1 weak motive or maybe 6 strong motives. And for a typical activator, the more motives you place, or the stronger motives you place, the more activity you get, and for a typical repressor, the more you place, less activity you get. However, biology is not that simple. We found quite a number of factors where this picture is much more complex. You place sites, let's take this one. You place sites, you get activity. You place more sites, it goes down again, and you switch from activity to repression. So this is now a factor that is both an activator and a repressor, and this is not some artifact of computing here, weird score. If we take high affinity binding sites, we put 1 high affinity binding site, we get an activator. We put more high affinity binding sites, we go to a repressor. If we take low affinity binding sites, initially we have like no protein bound. If we put a couple of sites, We go to an activator, put more sites, we have more protein, we go to a repressor. And this is really quite universal, so there are a couple of factors in our system that, you put more sites, you get more activation, so they are like activators. Others are repressors, but there's quite a number that have this duality behavior. And I think this is really something that is emerging in the community working on non-coding DNA, is that a lot of transcription factors have dual roles. And actually this survey here in yeast found that few TFs have dedicated activator and repressor roles, while many have this dual function. This is because many TFs have both activator and repressor domains. And there are different models on how this can be, on how this can work. And we advocate for transcription factors having incoherent effects on different steps in a transcriptional cycle. So here, if you think of a polymerase, or you can name those reactions as you want. Like you can think of it as a polymerase binding to DNA and then being released from pausing and then transcribing. And if these different steps are regulated by transcription factors in incoherent ways, you can initially have a rate-limiting step here that's activated by the TF, but once you increase the TF dose, then suddenly the effect of repression on another step in the cycle becomes dominant. So key message here, gene regulation is far from simple because transcription factors are not simply activators or repressors, they can be both. Once we start combining transcription factors, this gets even more wild. So here what you can see is two extreme examples where this factor on its own, it's an activator. In every cell state it activates. And this factor on its own also is an activator. It activates in every cell state. Those factors on their own never repress. However, if you combine them, you get activation in some cell states and repression in others. We have shown that this type of behavior depends on the distance between the binding sites. So it's only observed if the binding sites are fairly close together. And we have simulations from AlphaFold suggesting that there's direct protein-protein interactions involved where one factor sequesters the activation domain of the other, thereby making the effect of repression dominant. What this creates, however, are enhancers that are highly sensitive to ratios and transcription factor dose. We followed that up in detail by creating a lot of enhancers that had just binding sites for those two factors and varying the ratio and binding sites that they have, and then basically what you find is if one dominates, you get activation, and if you have both factors, you get repression. So to summarize this wild ride through transcription factor biology, there's these widespread negative synergies between activators where one activator may be quite unspecific. This is meant to resemble a landscape of differentiation of blood cells. So stem cells would be sitting here, and then you have different lineages. And this factor may be expressed in an unspecific manner a bit all over the place. And this factor too, and the activity actually resembles the expression pattern. However, if there's a strong negative synergy between them, you suddenly get repression in most of those cell states and activity only in cell states where one factor strongly dominates. And this is actually a very nice design principle of this biological system because it allows a very efficient repression of lineage genes in hematopoietic stem cells, if you think about it. So far the biology. Now let's get a bit into modeling and deep learning. I mean, I don't want to raise the hopes too much for leaving out the genome cross-validation since we've been here working with 38 transcription factors and the genome has 1,600. However, of course, we did this exercise of now taking differentially accessible elements, cloning them into a vector and doing MPA with them. And we also designed some more complex synthetic constructs which have quite a lot of transcription factor binding sites. And then we used a pretty much out-of-the-box deep model trained with all the data that I've shown you so far and tried to predict these two libraries. Performance on tenfold cross-validation on the trained data itself is that's what it is, which comes from the experimental assay. That's really a bit going into detail. The assay has certain noise, and if we measure noise-free data, we essentially get an R-square of 0.71. So I think this is like a better estimate of how good our model really is. But now comes the part that I actually wanted to show. When we then apply this to a complex synthetic library that has binding sites for the same transcription factors, the performance of our model is about as good as on the trained data itself. And if we apply it to natural genome, the performance is like 0 if none of our motifs are present. However, if we're looking at enhancers that have many motifs for our transcription factors, which probably conversely means that they don't have any other transcription factors, then the performance of these models becomes again as good or better as on the train data set that we used. So this fuels some hope that this hold out the genome concept can also work for cell state specificity and mammalian sequences. So the final part is that I want to go into a sequence designed by these models. They are not the most fancy AI. It's a black box machine learning model, deep model. So what we did is we formulated design tasks. And actually, we didn't make life easy for us because we said, okay, we have these 6 cell states here, and we try to achieve specificity to any combination of cell states. So there's 2 to the power of 6 different tasks that we gave this model. Then we initialized with some random DNA or some model-guided placement of transcription factors, and then did a couple of rounds of in silico evolution using our deep model as an oracle to score. And this produced then sequences that the model thought should work in a certain cell state specific manner. So this is an illustration of the design tasks. Red means on, blue means off. For example, this task here would be on in neutrophils, monocyte, and eosinophil precursors, and off in the remaining cell states. The model itself, or the oracle itself, has certain opinions on how feasible these tasks are, because certain combinations of cell types, let's say a stem cell, a monocyte, and a red blood cell, this is just something that the model thinks like it cannot do, right? So it doesn't produce any sequences that pass its thresholds. Whereas for other biologically more plausible tasks, there is a hierarchy to these cell types. The model produces many pieces of DNA that it thinks are great, and now this plot is a bit complicated. It essentially it's like a histogram. Each column is a histogram, and it scores how many of those DNA pieces that we synthesized and performed as expected. And you can see that for the more feasible task, we usually meet the design task in around 5 out of the 6 states, sometimes more. I see that this is not the most easy to read plot, so we made some box plots. The red boxes are, this is an aggregate of all the sequences we designed, and the red boxes show the activity in the cell states where the sequence was meant to be active. Blue shows activity in the states where it was meant to be inactive. So very, very good separation, and these are some example design tasks where, for example, we try to make DNA active in these 3 cell states, and that is the measurement across 21 sequences that were supposed to behave like this, and this is the measurement in the remaining cell states. So to close this, we did a systematic screen of minimalist enhancers, which on one side on the biology, we started this project a little bit more with the deep learning and AI and design aspect, but what we then really realized is that this is really powerful for learning a lot of things about transcriptional regulation, including things that we did not expect. But it is great training data, and especially when it comes to sequence design tasks, because what we saw is the variability of these synthetic enhancers is larger than the one of natural DNA sequences. So you can read this, was published recently, and now I don't know, we have, I've kept time well, so I think I have 5 more minutes. It's a bit off topic, since this is a mutational scanning symposium, but we have this other thing, which is epi mutations. So again, the motivation for this project is hematopoiesis. I told you we have 100,000 stem cells, and they are all unique, so we can think of them as clones, 100,000 clones, and as we age, some of these stem cells get a bit tired and disappear, and the others need to make up for it and expand. This is typical for aging. These stem cells have certain functional biases, and it's the first step towards cancer formation. But the problem is that these HSC clones are difficult to characterize in human, because how do we follow clones around without using genetic lineage tracing and without sequencing the whole genome of all these single cells? So in come epi mutations. Our DNA gets modified on the cytosine with methylation. And these methylation patterns get established at some stages during development, but then, of course, DNA gets copied over and the methylation pattern gets copied over as well. However, this is an error-prone process, so you accumulate epimutations. Problem, or the reason why epimutations haven't been used a lot for lineage tracing, or is in general something that people don't think about a lot, is that this is quite difficult to read out at the single cell level because you need readouts of single CpG methylation in single cells. And we have an assay for it. I don't want to go into detail. This is on the Mission Bio platform. Just want to say one thing. Mission Bio can do many things. It can measure DNA, so mutations, what you guys are interested in. I saw a couple of the talks just now and some posters outside. We're doing mutational scanning and trying to combine transcriptome with genome. And this is actually something that this platform can also do from the same single cell, so you can get RNA on top of mutation, on top of methylome. But the important thing for this talk is we do 1,000 CpGs, targeted methylation state in tens of thousands of cells, almost no noise because it's a targeted method. And then we did a ground truth experiment where we labeled hematopoietic stem cells with a genetic lineage tracer and read out their epimutational profile. And if we do a UMAP on this, and the previous speaker beautifully introduced what a UMAP is, we see that these cells cluster by two things. They cluster by the clone. So all of these red cells here come from the same mother stem cell. All of those cells here come from the same mother stem cell. But they also cluster by what they are doing, whether they are like red blood cell precursors or white blood cell precursors. And interestingly, these two processes, clone and differentiation, map to different CpGs. So we have one set of CpGs that we call dynamic, and if we do a UMAP on the dynamic CpGs, we get differentiation. But if we look at the other CpGs, the ones that are passively maintained and that rather suffer from epimutations but not from active regulation, we get a UMAP that, well, does not reflect differentiation state, but rather reflects clone. This was mouse for ground truth, but we went on to use this in human, where it also works, and you can see that young individuals, this is a 22-year-old, it's just a blob, there's no structure. The older the individuals get, just ignore this one, he had too few cells, don't see anything, but the older the individual gets, the more structure there is, and what this structure is essentially a benign tumor of the blood, because it's like a clonal expansion of cells. So to summarize, just brief vignette, because it's a topic that we work in and that I'm quite fascinated by. Epimutations have a much faster rate than DNA mutations. They are great for lineage tracing, also in human, if one can read them out. And scary, everyone aged 50 has benign tumors of the blood. You can read about this in 1 hour. With that, I would like to thank you for your attention, and I'm welcoming questions.